Olfaction. Olfaction, or the sense of smell, is the ability to perceive volatile molecules called odorants that are dissolved and detected by chemoreceptors inside the mucus of our nasal cavity. The olfactory organ, which is the organ of smell, is composed of the olfactory epithelium, which lines our superior region of the nasal cavity, lamina propria, areolar connective tissue, and mucin-secreting olfactory glands. The olfactory epithelium has three types of cells. The olfactory receptor cell, these are neurons that detect odors. The olfactory receptor cells have a single dendrite. Projecting from the dendrites are the olfactory hairs, which extend into the mucus layer. The olfactory hairs contain these chemoreceptors in their plasma membrane, and they detect about 1 out of 400 specific types of smells that we can perceive. The axons of these receptors are then forming into the olfactory nerve, cranial nerve number 1, that pass through that cribriform plate of the ethmoid bone and goes directly to the olfactory bulb. And on average, between every 40 to 60 days, the olfactory receptor cells are regenerated. The cilias do protrude downward into the nasal cavity, and the axon protrudes upwards as synapse towards the mitral cells. These cells help relay the signals towards the olfactory nerve. So odorants are over here on the bottom. We have the supporting cells shown here, the olfactory sensory neurons, and then we have the ethmoid bone, and then we have the glomerulus, eventually the mitral cells, which will then relay the action potential through the cranial nerve number 1. Detecting smell and olfactory transduction. So during normal, relaxed breathing, most of our inhaled air does not pass over the olfactory epithelium, so we must sniff up or breathe deeply. The odor molecules will then diffuse into the mucosal layer where we have the olfactory receptor cells. The soluble proteins within the mucus of the nasal cavity help bind to these odorants. The kind of compounds that bind to those odorants are called the odorant binding proteins. So this is a G protein cascade. Stimulation of olfactory receptors does activate G proteins inside these cells. It begins by activating a G protein. The GDP is bound to alpha subunit of the G protein, is then exchanged for GTP, and activates and breaks off the alpha unit. The alpha unit will then target an effector enzyme called adenyl cyclase. Adenyl cyclase will take ATP in our cells and convert that into a second messenger called cyclic AMP. Cyclic AMP then binds and opens up cation channels that allow sodium and calcium to rush into the cell. There then, as a result of the entering of sodium and calcium, there's a local potential that's generated within the olfactory hairs, and that creates an action potential along the receptor cell. The binding of the neurotransmitters by the secondary neurons results in the glomeruli sending signals through the olfactory pathway to various parts of the brain. The primary olfactory cortex inside the temporal lobe allows us to consciously perceive the smell. The hypothalamus plays a role to control the visceral reactions such as salivation, sneezing, or even gagging. And of course, the amygdala is a place for center recognition of odors, and it relates it directly to memory. The olfactory tract are paired axon bundles of the mitral and tufted cells, and they project all the way to the inferior frontal lobe, to the primary olfactory cortex of the temporal lobe, as well as the hypothalamus and the amygdala. Gustation, which is a sense of taste, occurs within our gustatory cells, or taste buds, and whenever they come into contact with taste-producing molecules inside our food or drink. The papillae are these epithelial and connective tissues elevated surface of our tongue, and there are 4 different types. We have the short, spiky, bristle-like papillae that serve as mechanical function in the anterior 2/3 of our tongue, and these ones do not house taste buds, but they do detect texture of food. These are called the filiform papillae. We have the block-like mushroom-shaped papillae that contains only a few taste buds, and these are located at the tip and the sides of the tongue. These are called the fungiform papillae. And then the largest papillae, but the fewest in number, are the majority of where taste buds are housed, and these are called the valiate papillae. And the last one are papillae that are found primarily during infancy and early childhood, and they house only a few taste buds. These are called the foliate papillae. The gustatory cells within the taste buds are these specialized neuroepithelial cells, and the dendritic endings of each of these gustatory cells form the gustatory microvillus, or the taste hairs. Many taste microvilli extend through an opening in the taste bud, called the taste pore, to the surface of our tongue. So compounds that will then trigger taste are called tastants. They dissolve in our saliva, and they allow for the stimulation of the gustatory receptors through those little taste pores. Don't forget that these gustatory cells are chemoreceptors. There's also transitional cells that help support cells between the taste receptor cells. Discrimination and the physiology of taste. There are 5 basic sensations that no longer are mapped as specific locations on the tongue. Sweet taste sensation is produced by the organic compounds such as sugar. The salt taste sensation is produced by metal ions such as sodium and potassium. Sour taste sensation is produced by the presence of hydrogen ions due to ingestion of acids. The bitter taste sensation is produced by alkaloids such as quinine and unsweetened chocolate, nicotine, and caffeine. And then the last one is umami taste sensation, which is attributed to amino acids like glutamate, aspartate, and they produce a savory or meaty flavor. Some research has shown that there are sensory receptors that detect fatty acids and other possible tastes like alkaline, metallic, and water-like. Let's talk a little bit about the tastings and how they bind to specific receptors in the plasma membrane of the gustatory cells. They then trigger depolarization in several different ways. So first, sweet, bitter, and umami. Sweet, bitter, and umami, they activate G proteins as a secondary messenger to cause depolarization, and thereby increasing calcium ion inside the cell that leads to depolarization. On the other side, salt and sour and some umami chemicals depolarize the cell directly by altering the ion channel. These ones over here on the right directly change the ion channel. These depolarizations generate a local receptor potential that initiates the nerve signals that cause the release of the neurotransmitters on its basal side and stimulates the primary neuron. And that will then trigger either the facial or the glossopharyngeal nerve to send signals towards the brain. So directly when a compound goes, when one of these ions goes in, it increases the calcium and then depolarizes. These guys are all GPCR or G protein couple receptors. Over here is a pathway showing you how each of these work. Salty occurs when sodium from salty foods enter through the sodium channel. This will then cause depolarization, leading to the opening of voltage-gated calcium channels, and therefore more calcium rushes in, triggering an increase in the membrane potential, leading to the release of neurotransmitter of glutamate. In sour, the protons from the sour fluid block the potassium channel, therefore potassium gets trapped inside the cell. Because potassiums are trapped inside the cell, it causes a change in charge in the membrane, making the inside more positive, causing calcium to rush in more, and then causing the neurotransmitter to be released, stimulating the nerve. In a sweet taste, a sweet substance binds to its receptor, causing a G protein to activate. The activated G protein, also known as gusducin, activates adenyl cyclase, the effector enzyme, which will convert ATP to cyclic AMP. And cyclic AMP activates a protein kinase that then phosphorylates and closes the potassium channel, therefore causing the membrane potential to increase. Calcium comes in and causing neurotransmitter to release. In the case of bitter, a bitter substance will bind to its receptor, causing conformational change, again activating a G protein. In this case, the G protein is called transducin. Transducin is going to activate the effector enzyme phospholipase C, called PLC. That will then activate the second messenger PIP2 and inositol triphosphate, IP3, which will increase the calcium inside the cell from the smooth ER, causing a change in charge of the membrane, causing the neurotransmitter to be released, and then a signal goes through the nerve.